Cloning vectors for plants and animals. This topic is taken from the chapter of Biotechnology Principles and Processes. TI plasmid, which is tumor inducing plasmid, refers to the plasmid of Agrobacterium tumefaciens. So, Agrobacterium tumefaciens is a pathogen of a dicot plants. So, it is a pathogen of a dicot plants. Then it is a tumor inducing plasmid. It will deliver a piece of a DNA which is also known as tDNA. A tumefaciens is a plant pathogen. It produces tumors in the plant it infects. So tumefaciens, that is Agrobacterium tumefaciens, is a bacteria which will causes diseases, because it is a pathogen. It will produces the tumors that spread and it will infect the plants. tDNA will transfer the normal plant cells into the tumor cells, and these tumor cells will produce the chemicals required by this pathogen. TI plasmid can be modified into a cloning vector by removing the genes responsible for pathogenicity. So, tumor-inducing plasmid will be modified into a cloning vector. How they will be modified into a cloning vector? By removing the genes which are responsible for pathogenicity, means the genes which are responsible for causing diseases are removed, and this TI plasmid is modified into a cloning vector. Retrovirus, these are the virus that infect animals. Now the agrobacterium case is finished. Now the retrovirus. These are the virus that infect animals. They produce cancer in animals. So this retrovirus is the virus that will infect the animals. They will produce cancer in the animals. These are the cancer cells. They are very harmful. Retrovirus is the harmful virus. They will infect the animals by producing cancer in the animals. Retrovirus can be disarmed to be used as a cloning vector. Similarly, retrovirus in the animals have the ability to transfer the normal cells into the cancer cell. It will transfer the normal cell into the cancer cell. As in the TI plasmid, we modified the cloning vector by removing the genes. Similarly, retrovirus is also modified into a cloning vector by removing the genes which are responsible for the producing cancer. Quick recap of cloning vectors for plants and animals. So, Agrobacterium tumefaciens is a pathogen which is present in the dicot plants. This is able to deliver a piece of a DNA which is also known as tDNA to transfer the normal plant cells into the tumor cells. So it is the plasmid. Okay, and there are tumor cells which will produce the chemicals required by these pathogens. So tumefaciens is a plant pathogens which will infect the plants. Now that these TI plasmid of the tumefaciens are modified into a cloning vector by removing the genes which are harmful, which are responsible for the pathogenicity, means they causes disease. Retrovirus it will infect the animals. Retrovirus is a virus that will infect the animals, and this will produce cancer in the animals. Now similarly, retrovirus are also modified into a cloning vector by removing the genes which are responsible for the production of cancer in the animal.